Good afternoon, everyone. I am Kimberly Grinston, Executive Director for the Missouri Board of Pharmacy, and welcome to the board's webinar on the recent update to Missouri sterile compounding regulations. We have a lot of information that we're going to share today. Several people will be participating in the webinar today, including myself, our sterile compounding inspector, Katie DeBold, and also, of course, our chief inspector, Tom Galinsky. We're going to give you just a little information about how the webinar is set up, and then we'll talk to you a little bit about what the board did and why we're actually hosting this webinar today. And then Katie's going to give you more specific information on what changes were made in the sterile compounding rule and what you may look forward to with the amended rule. And then after that, we'll have an opportunity, hopefully, to answer some questions for today. Now, the webinar, we have quite a bit of presentation today, and hopefully we'll be able to get to as many questions as possible. Tom and Katie will tell you how to answer a question, but we're going to ask that you can submit questions now, but we'll answer questions at the end of the webinar. Again, thank you for attending today. I want to remind everybody that there are resources on the board's website that will give you more information about the changes to the sterile compounding rule. But before we go into more information, I'll introduce Katie and have her tell you how our webinar is set up.
Good afternoon, everyone. As Kim said, my name is Katie DeBold, and I will be presenting the webinar today. So just a little housekeeping, if you haven't been to one of our webinars yet. All of the participants will be in the muted listen-only mode, and if you're having problems logging in and hearing the audio, you can also dial in by telephone. This webinar is board approved for 1.5 hours of live pharmacist continuing education. You must be officially signed up and logged in via your computer. If you are watching with a group of people, only one person will get the board approved CE, and that's the person that's logged in. So the CE credit will not be issued to those that are listening over the phone. At the end of the survey, we ask that you complete a post-survey within 48 hours, and we'll tell you how to do that towards the end of the webinar. And just a reminder that CE credit is not submitted to the CPE monitor. Instead, we will be sending you a certificate. There are copies of the handouts posted on the board's website, and this webinar is also being recorded. You'll be able to find the webinar hopefully within 30 days or sooner, and it will be under the Publications Resources link and probably on the first page as well. You do not get CE credit for watching the recorded version, only the live version. Feel free to ask questions throughout the webinar. As Kim said, we will be answering questions at the end, so just type in your question in the question box, and we will hold on to those for the very end. So before we get started, I just want to provide a little background about myself. Like I said, I'm an inspector with the board. I have been with the board for about 9 months now, and I was hired on specifically to provide a specialized focus for sterile compounding within the state of Missouri. Before this, I was employed mostly in the hospital pharmacy world, and that is where I was able to gain my experience and passion for sterile compounding. So webinar objectives, today we're going to review updated requirements of the emergency rule, which was effective August 4th, 2016, and we also be reviewing updated requirements of the amended rule, which we anticipate to be effective in March of 2017. And we'll explain the differences between the emergency rule and the amended rule as we move forward. I'll also be answering questions at the end of the webinar. So now I'd like to introduce back Kim. She's going to give us a background behind the process of the emergency rule.
Well, I just have a few questions that I wanted to answer, and I think one of the questions that we've heard at the office is why now? As you know, the board has been looking at the sterile compounding rule. For those of you who have read the sterile compounding rule recently, you know that the sterile compounding rule has a lot of revisions that needed to be made prior to the actual emergency amendment. After NECC, the new Compounding Center, the board began looking at our sterile compounding rule to see what, if any, changes needed to be made. Many of you participated in a series of public board meetings where the board actually drafted a comprehensive revision and redraft of the sterile compounding rule. Actually, the board was proposing to completely rescind the rule and repromulgate it to update it to 797, or to make it more similar to 797. In the interim, 797 introduced its draft revisions, and so the board took the rule under advisement just until 797 was finalized. The thought was that the board wanted to incorporate the newest standards and not publish a rule that would be once again a version behind 797. Actually, now we're several versions behind. After we hired Katie here at the board office, and she's really been an asset to what we do with the board, we received just different notices, and we found different information during inspection about sterile compounding activities in this state. As you know, sterile compounding is probably the highest risk activity performed by our Missouri licensees. In response to what we saw, not only on the national level, and in response to FDA recall notices, some affecting licensees that were non-resident licensees licensed in Missouri, and also what we were seeing on inspection. The board voted to proceed with an emergency rule that would address some of the compliance issues we were seeing at the time. So the goal was, again, not to comprehensively revise the sterile compounding rule. It still needs a lot of updating and simplification. But the goal was to give board inspectors and the board the tools they needed to enforce the minimum levels of protection. And I'd like to stress that. Everyone knows that the mission of the board is to protect the public. That's what the board does every day. That's what our inspectors do every day. And we found ourselves looking at our rule and unable to enforce some of our minimum patient protection standards that were standard nationwide. Some of them came from 797, but we realized that the board's rule really needed to be updated on an emergency basis. So what the board did is, instead of doing a comprehensive rule review right now, we understand that 797 is still being revised. The board voted to, again, adopt just those emergency provisions needed to address what was happening in the sterile compounding rule right now. Our goal was, or the board's goal was, to protect the public by establishing those minimum safety standards. Next question, did the board adopt USP 97? And a lot of people have asked, why doesn't the board just adopt 797? And it's a good question. As you all know, 797 is the national standard and probably is the standard that all sterile compounding pharmacists and pharmacies are familiar with. The current version of 797, and understanding it's going through revision, is not written in regulator language. And so the current version has a lot of language that is unenforceable. For those of you who've had the pleasure of going through 797, you'll notice that the language includes things like recommendations and language that says that licensees should, but it doesn't say what is required and what's not required. In reading the current 797, it was very hard to simply adopt the rule. I always tell this, I went to a state meeting of all 50 states, and one of the states stood up and said, we would be glad to adopt 797 once we figure out what's required. That was one of the struggles we had with the current version. Additionally, in addressing the emergency need that we saw during inspection and in light of the FDA recalls, the board knew that a comprehensive revision or adoption of 797 would result in significant costs for licensees and our pharmacies, which could detrimentally impact sterile compounding supply in this state. And so the board chose not to adopt 797 at this time. Of course, 797 is going through a revision. It has gone out for comment once. I understand thousands of comments were received. The board submitted comments as well. And we understand that it will continue to be revised, and we don't anticipate an effective date before 2017 and possibly as late as 2018, if not later than that. So the board has not adopted 797 at this time, but they intend to at some point, and I'll discuss that in just a moment. Let me tell you about the emergency rule versus the amended rule. Under Missouri law, a state agency can promulgate an emergency rule to address a significant patient harm that is present. And so looking at, again, all the information before the board, that is what the board did. Emergency rules become effective in 10 days, which is the reason for the August 4th early effective date. An amended rule, when you file an emergency rule, you also have to tell people what the permanent rule is going to be, and that would be the amended rule. The amended rule in this case is very similar to the emergency rule with some key differences that Katie's going to discuss in a little bit. But the emergency rule was effective August 4th, and the amended rule will be effective, we believe, in March of 2017. Both copies of the emergency and amended rule will be on the board's website. The amended rule is there now. The emergency rule, is there now. The amended rule is still being drafted. The final form, it's been approved by the board, filed with the Secretary of State's office, but the official publication version is still being edited for commas and punctuation and those things, and that will be available shortly. We will post it as soon as we have the official copy from the Secretary of State's office. We anticipate that happening within the next week or so. What are the board's future plans? The board does intend on looking at the sterile compounding rule again once 797 revisions have been completed. So this is not the last revision, and I want to make sure I stress this for all licensees. The emergency rule addresses just the compliance issues that the board was presented with today. It addresses, again, the emergent need. The amended rule goes just a tad bit further on things that the board thought should be in place long term, but the board will look at the rule again and will likely adopt all, if not significant portions of 797, depending on what the final language says. So you will see that coming in the future. Please monitor the board's website, our newsletters, our e-alerts, because we will provide information on what's going to happen and also give you an opportunity to comment on the revision of 797. One of the things I want to stress is that the board, in looking at this draft, intentionally looked at fiscal cost to licensees. Again, the thought was we did not want to promulgate anything that would drastically affect the sterile compounding supply. So you will see no facility or structural changes in the current draft, but I think those changes will be coming in the next draft, again, after 797 is revised. That's just a little introductory information about why the board did it filed an emergency rule, what we intend on doing after the emergency rule is filed, and now we're going to get into more of a discussion on what the emergency rule and amended rule actually require. So, having said that, I'll turn it over to Katie.
Thank you, Kim. So now I would like to review who this rule applies to. This rule applies to all Missouri Board of Pharmacy permit holders that have Class H, which is sterile compounding permit, and or Class E, which is nuclear pharmacies, on their license. Is this rule applicable to hospitals? The answer is it depends. If your hospital is doing sterile compounding for inpatient use only, this rule does not apply to you. That is still regulated by the Department of Health and Senior Services, so they will be regulating all sterile compounding for inpatient use. If you are a hospital that holds a Class B and H permit with us, and you are doing sterile compounding for outpatient type activities, then this rule would apply to you. Some examples of outpatient type activities include compounding TPNs or antibiotics that will be sent home with the patient, or if you have an off-site infusion center, depending upon where that's located, that could be considered an outpatient type activity. So inpatient activity only, this rule does not apply. Outpatient type activity, it might apply depending on your situation. So the way I've designed this webinar is that I've taken up the emergency rule and I've broken it up into different sections. So we will be reviewing each section and I'll explain the changes associated within. So if you have the emergency rule sitting in front of you, you should be able to follow along sequentially. So the first thing that people worry about whenever they hear that there is a change to sterile compounding regulations is what sort of construction am I going to have to do? What sort of redesign am I going to have to do with my facility? And the answer to that question is nothing right now. Neither the emergency rule or the amended rule have changes to any facility or structural design. Most of the changes are operational changes. And some of those operational changes include training and media field testing, increased garbing requirements. We provided some clarification regarding controlled areas versus buffer areas. We have increased cleaning and disinfection requirements. We plan to implement an environmental sampling plan, which will be in the amended rule. And we also provided licensees with guidance about how to perform remedial investigations. So the first part of the emergency rule, of course, deals with definitions and terminology. So I thought I would start out with one of the more important definitions, which is what is a compounded sterile preparation. So throughout the years, the board has gotten questions about what sort of dosage forms need to be prepared as a sterile preparation. So we included a list of some of the common dosage forms that need to be prepared as sterile. This is not an exhaustive list, but it at least gets you started. So we have baths and soaks for live organs and tissues, epidural and intrathecal solutions, bladder and wound solutions, injectables, IV solutions, parenteral solutions, implantable devices such as pellets, for example, inhalations, irrigations, ophthalmic preparations, if you are repackaging any sort of sterile preparations, and also the assembly of point-of-care systems. And if you're not familiar with point-of-care systems, I will review that later on in the webinar. One question that we get a lot is regarding nasal sprays and nasal irrigations, if they need to be prepared as sterile preparations. And we have answered that in this rule, and the answer is no, they do not have to be prepared as sterile. So moving on with the terminology changes, we've replaced the Class 100 and Class 10,000 terminology with ISO classifications. Class 100 and 10,000 are, of course, some outdated terminology, so they will be updated with ISO Class 5 instead of Class 100 and ISO Class 7 instead of Class 10,000. We added the definition of a buffer area. So a buffer area is an ISO classified area, which is ISO Class 7 or better, where your primary engineering control is physically located. So your primary engineering control is going to be your laminar airflow workbench, biological safety cabinets, and what people typically refer to as isolators. Those are all considered primary engineering controls. So the room that is physically located in, if it's ISO classified 7 or better, you would refer to that area as a buffer area. So for those pharmacies that do not have an ISO classified area, we came up with the definition of controlled area. So a controlled area is a room or area designed for sterile compounding. The area is separated from other activities and operations by a line of demarcation. And that line of demarcation is a way for pharmacies to help define their sterile compounding space within their pharmacy, since they don't have that ISO classified area. It also serves as an operational tool to help assist your staff so that they know once they've crossed this line, they need to treat that area different. So they need to garb, clean, maybe do some different material handling things at that point. In addition, we also removed the terminology of isolator. The definition of an isolator is that it's actually a type of primary engineering control that is used often in pharmaceutical manufacturing, and it has an automated system that's built in that distributes a sporicidal chemical agent, and that is used for a vaporized decontamination process. So that is rarely seen in pharmacies, so we decided to get rid of that terminology since it didn't really apply to our compounders, and we have replaced it with the phrase RABS, which stands for Restricted Access Barrier System. So this is the new terminology that I'll be using throughout the webinar, and throughout the rule you'll see it as well. So this is what people will typically refer to as glove box or isolators. So now we'll be calling them RABs. It includes both CAIs and CACIs. So CAI stands for Compounding Aseptic Isolator, and those are used for non-hazardous compounding, whereas CACI stands for Compounding Aseptic Containment Isolator, and that's used for hazardous compounding. So the way that I remember the difference between those two is that the compounding aseptic containment isolator, the one that has the extra C in there, if you think about when you're compounding with hazardous drugs, you always want to contain your hazardous drugs. Therefore, your CACI, the one with the extra C in there for containment, will be used for your hazardous compounding. So still within the definition sections, we've come across risk levels. The risk level stratification is mostly unchanged. We did decide to remove the administration time from the risk level definitions. And this may allow some pharmacies the ability to change their risk levels. So previously, when you're determining risk levels, there were kind of 4 different parameters you'd have to look at. The first would be what were your starting materials and supplies. If they were non-sterile, you would automatically be put into the risk level 3 category because you're starting with non-sterile and you're sterilizing that preparation through compounding. Next, you would look at what sort of compounding activity you were performing. So if you were doing any batching without any preservatives or using an automated compounding device, then you would be considered a risk level 2 category. If neither of those applied, you would then look at your storage conditions and the dating that you are assigning your preparations, in addition to the administration times of your preparations. So previously, if your administration time of your, say, epidural was going to be running for greater than 48 hours, so maybe over 3 to 4 days, you would automatically be put into the risk level 2 category. Now that the administration time is removed, you no longer have to worry about those administration time limits. So the dates are still the same. In order to be considered a risk level 1 category, you have to have a beyond use date of 48 hours or less when you're storing it at room temperature. If it's stored in the fridge, your beyond use date must be 7 days or less, or if it's frozen, it must be 30 days or less. Anytime you exceed those beyond use dates, you will be put into risk level 2, and you'll have to follow all of those regulations. Section 2, policies and procedures. There weren't really any changes within this section. Rather than provide a whole list of what policies and procedures are required, we decided to incorporate them into different sections throughout the rule. So I will make sure to point those out as we go along. And I just wanted to remind everyone that an annual review of sterile compounding policies and procedures is still required, and you want to make sure to document that. Section 3, personnel education, training, and evaluation. So we've updated our training requirements to include more specific competencies. Our inspectors have found that training programs varied greatly among licensees, and we thought this was an area that could be improved upon. So our previous rule has the terminology of suitable, didactic, and experiential training. And suitable training is, of course, a subjective term. So we wanted to provide some objective competencies that everyone would have to meet before they were able to start sterile compounding. And those competencies are referenced in Section 10 of the rule, which I will discuss later on in the webinar. Now, that's applicable to all risk levels. If you are a risk level 2 or 3, you will have to add into your competencies some competencies that are specific to your risk level. So, for example, if you're risk level 3, you'll need to have some additional competencies for sterilization or end preparation testing. Additional training might be required for changes in risk level or compounding methods. So if you get a new TPM compounder or you get some new technology that everyone needs to be trained upon, that will need to be done and documented appropriately. And this is one of the areas that you will need a policy and procedure on, is for staff training and assessment. So Section 4 is storage and handling. This is mostly minor changes to update or clarify the language. You'll notice a phrase within the storage and handling section that talks about maintaining chemical and microbiological stability, and you'll see this phrase throughout the rest of the rule as well. The reason that we added this phrase is to remind our licensees that you should always, when you're going through the storage and handling and beyond use date process, you should always consider not only the chemical stability but also the microbiological stability of that preparation. So what risks of contamination occurred, and you have to use whichever would be shorter. Other changes include an addition of daily incubator temperature monitoring. If you have an incubator, you need to make sure that it's working properly and document that. And then we also added a reference to Section 21 regarding recalls. Section 5 is facilities and equipment. We have completely rewritten this section. We felt the past section had a variety of regulations that needed their own separate section. Cleaning, for example, needed its own section. So throughout Section 5, you will see mentions of those sections, and it will reference you as to where in the rule you can find those removals. So now I'd like to review some of the existing facility requirements. Since we haven't made any changes to physical structure or room design, we'll go ahead and review the already present restrictions that are in place. So for risk level 1, preparations must be prepared in a primary engineering control, which is located in a controlled area, which is defined by a line of demarcation. So your primary engineering control can be put into a non-ISO classified area, as long as you define that area with a line of demarcation. Risk level 2, preparations must be prepared in a primary engineering control, which is located in ISO class 7 buffer area, or if you are using a RABS, so remember RABS is the new terminology for isolator. If you're using a RABS, it should be located within a controlled area, and again, since it's a non-classified area, you would need a line of demarcation. And the same is true for risk level 3. So a PEC must be an ISO class 7 area or a RABS with a controlled area with a line of demarcation. So still continuing on in facilities and equipment, you'll see that the cleaning requirements have been moved to their own section. So that'll be section 17, which we'll review later on. You will notice a sentence within the risk level 3 section which talks about risk level 3 preparations shall at a minimum remain risk level 3 for the life of the preparation. And I just want to explain what that means. So risk level 3 compounders, oftentimes they will prepare a big bulk solution. That bulk solution is started to be compounded with non-sterile materials and equipment, so they will do all the proper sterilization. They will send off that compound for sterility testing, endotoxin testing, and once they have proven the sterility of that, they will then use that bulk solution to prepare final preparations. So those final preparations are still considered risk level 3 compounds. Even though that bulk solution has been proven sterile, it doesn't change the fact that the preparation was originally compounded with non-sterile ingredients. So the final preparation and the bulk solution are both considered risk level 3 preparations. For automated compounding devices, they need to be calibrated initially to use and also on a daily basis. So they need to be tested for content, volume, weight, and accuracy. A pharmacist needs to be reviewing those test results, and that needs to be documented within the pharmacy's records. We added a requirement for pressure differential monitoring, so there's no requirement to install a pressure monitoring device. However, if your pharmacy currently has one installed, the results must be monitored and documented daily. A continuous monitor is acceptable as long as that system is able to maintain documentation of the alerts and recordings throughout the day, and that does need to be reviewed by a pharmacist on a daily basis. So moving on, still within Section 5, we'll talk about certification. So the frequency of certification is unchanged. All of your primary engineering controls and ISO classified areas must be certified initially before they are used and every 6 months thereafter. In addition to the normal certification requirements, you also may need to do some recertification. Recertification must occur if there are any major changes or services to your PEC or ISO classified area. So if you are replacing a HEPA filter or some sort of blower on your piece of equipment, you need to have it recertified. If your PEC is relocated or if you change the structure of your ISO classified area, you will need to recertify your primary engineering control and your ISO classified room. Your certification results must be reviewed by a pharmacist, so make sure that your certifier is getting you a copy of those results and make sure that you understand your certification reports and what is actually on there. It is your responsibility to understand how your equipment in your room is operating, so that way if there's a problem or a failure, you can be able to investigate that and correct it further. So that certification result review must be documented within the pharmacy's records. If you do discover a deficiency or a failure, like I said, you need to investigate that and correct it prior to further compounding. So during that investigation period, you might need to limit the use of that primary engineering control. You may need to initiate a recall, and you need to work through and solve the problem prior to any further compounding. Section 6 is a new section, Primary Engineering Controls. Is to provide guidance on the proper use of primary engineering controls. So your primary engineering controls must be located out of high traffic areas and away from conditions that could adversely affect their operation. So we're talking about fans, ventilation systems, any cross drafts. You don't want to keep fans within your controlled or buffer area as it can disrupt the primary engineering control's ability to maintain ISO Class 5 conditions. If you have a ventilation system or something that is not easily moved, I would recommend having your certifier do a smoke study to ensure that that your primary engineering control is still maintaining ISO Class 5 conditions while that ventilation system is running. Primary engineering controls shall be able to maintain ISO Class 5 or better, which of course is demonstrated on certification by your particle counts, so make sure that that stays up to date. We also need to establish recovery times for our primary engineering controls and identify what that recovery time is within the pharmacy's policies and procedures. So the recovery time kind of means two different things. If you have a RAB, so remember that's the new technology for isolator, you need to come up with a recovery time of how long it takes your primary engineering control to reestablish ISO 5 conditions while you're doing material transfer. So if you've used a RABS before, you know you have to open the antechamber, you have to put in your supplies, you have to close the door. So you need to establish a recovery time after all of that has happened in which the primary engineering control is back to normal operating conditions. And that is usually identified by the manufacturer of the RABS. They've usually done that testing ahead of time. For those that don't have RABs and have more traditional primary engineering controls, so laminar airflow workbenches or biological safety cabinets, the recovery time would be, an example would be if you turn off your PEC for whatever reason, how long it would take your PEC to get back to normal operating conditions once you've turned it on. Although I do want to point out that it is best practice to leave your primary engineering control running at all times to avoid any microbial contamination. Section 7 is a new section for controlled areas, and it helps to explain some of the expectations that we will have for controlled areas. A lot of these also apply to buffer areas as well. So some of the controlled area requirements include, for your non-classified areas, like I've said before, you need that line of demarcation to designate the area used for sterile compounding. Within that line of demarcation, the area must be clean, well lit, and free of infestation. If you have trash within that line of demarcation, it needs to be disposed of at least daily. For your ISO-classified areas, if you have furniture, carts, supplies, or equipment, it needs to be cleaned and disinfected before entry into the ISO-classified area. And just a word about carts is if you're using carts, it's always better to keep one cart in the ISO-classified area and then another cart on the non-ISO-classified area, because every time you move that cart out of the ISO-classified area, you'll then have to re-clean it before it's able to enter back into the ISO-classified area. All personnel entering controlled or buffer areas need to be appropriately garbed, which I will go over on the next slide what that entails. Some items that are prohibited in the controlled or buffer areas include tacky mats. Food, gum, eating, drinking, and smoking are all prohibited. You want to avoid having any particle shedding items in there, such as pencils, wood, corrugated cardboard, paper towels. Cotton items such as gauze pads are all high particulate generating items, and they should not be in that area. I've included a picture of the corrugated cardboard to show you the kind of cardboard that we're talking about. The corrugated cardboard harbors a lot of mold and spores, and they generally generate a lot of particles, so we want to avoid putting those in the controlled or buffered areas. The kind of waxy cardboard that your vials often come in, that sort of cardboard is okay to be in the controlled or buffer area. You don't want to bring any shipping or external cartons into your controlled or buffer area. They've been all over the place and they're probably very dirty, so we don't want those in your controlled areas. And you also don't want to store any non-essential supplies or equipment within your sterile compounding areas. You don't want to use it as storage space, which would just allow dust and particles to accumulate in that area. So Section 8 is garbing and hand hygiene. There were a lot of changes to this section, especially for our risk level 1 compounders. So as you can see before, our risk level 1 compounders didn't have any garbing requirements. That has changed, and now the risk level 1 compounders need to don non-shedding gowns, a hair cover, face mask, beard cover, and gloves. For risk level 2 compounders, we added the addition of shoe covers and the addition of sterile gloves. So the garb that risk level 2 people need to be wearing is non-shedding gowns, hair cover, face mask, beard cover, shoe covers, and sterile gloves. The only addition within risk level 3 is that the gloves need to be sterile. So risk level 2 and risk level 3 are actually have the same garb. I just wanted to point out that there are no exemptions for the compounding personnel that use the RABs. You need to garb according to your risk level. So even if your manufacturer says that they recommend certain garbing for the use of your RABs, you need to use the garb that is according to your risk level. And talking about RABs, if you are risk level 2 and 3 and you're using sterile gloves, well, you have to use sterile gloves with the new regulations. Those sterile gloves need to be donned over the RABs gloves. So what you're going to do is you're going to have your package of unopened sterile gloves. You're going to put it into your antechamber, put your arms into your RABs, and then don the sterile gloves while you're inside of your PEC that way. And just a reminder that all risk levels need to disinfect their gloves before use and frequently thereafter. Section 9 is aseptic technique and preparation. So within this section, we've given more specific requirements for hand washing. So you want to wash your hands and forearms for 30 seconds under warm running water. You need to remove your debris from underneath your fingernails. The amended rule includes the use of a disposable nail cleaner. So that the amended rule, again, is the rule that will be effective, we think, in March of 2017. So you'll have to add that piece of equipment onto your hand washing regimen. And there were also some other clarifications within Section 9 that added things such as all supplies, equipment, surfaces of ampules and vials shall be disinfected before going in your primary engineering control. And that can be done using sterile alcohol or a disinfectant that does not have a residue associated with it. For the risk level 3 section, we added that sterilization methods need to occur in a method that is recognized and confirmed by USP, and that sterilization method needs to be confirmed by a USP recognized sterility test. So the concept of in-use time is something new for our licensees, and we've included it here in Section 9. So the definition of in-use time is the time or date before which a conventionally manufactured product or a compounded sterile preparation, CSP, must be used after it has been opened or needle punctured. So all vials and containers must be dated and timed after that initial needle puncture has occurred. So the in-use time has no effect on the final preparations beyond use date. It's simply the amount of time that that specific vial or container can be used during the compounding process. So you might be asking yourself, how do I decide which date to put on my vial? You need to look at your vial or your package insert, and it should note on there whether your vial is a single dose vial or a multiple dose vial. The difference between the two is that multiple dose vials have an antimicrobial preservative added to them, and the manufacturer does all sorts of testing to prove that this preservative is effective, whereas the single dose vials do not have any additional antimicrobial preservatives added to them. So the date for single dose vials or containers is going to be a maximum in-use time of 6 hours unless otherwise specified by the manufacturer. So in order to get the manufacturer information, they'll either list that on the side of the vial or sometimes they will put it in the package insert. If their timeline is greater than 6 hours, it means they've done extra studies to validate that. And the reason that 6 hours is chosen is because once a microorganism is introduced into a new environment, it has a lag time of 4 to 6 hours before it starts replicating. So that's kind of a little background about where the 6 hours timing came from. For multiple dose vials and containers, the maximum in-use time is going to be 28 days, and of course that's much longer because of the preservative, again, unless it's otherwise specified by the manufacturer, and the manufacturer has done extra studies to prove that. Ampules must be used immediately and should never be stored, so they don't get an in-use time at all. So Section 10 is the aseptic technique skill assessment. You'll notice that the process validation section was removed and replaced with this one. The aseptic technique skill assessment is used to verify your aseptic competencies of all your sterile compounding personnel. It consists of both media fill testing and also direct visual observation of the following competencies. So these are those specific competencies that I was referring to earlier in the presentation. So you need to ensure that your employees are able to demonstrate proper aseptic technique and work practices. So this is going to include things such as avoiding touch contamination or using first air properly. Your employees need to demonstrate that they can clean and disinfect properly. And this is also true for non-pharmacy personnel. If you have environmental services or someone else that's coming in to do your cleaning, they need to be trained and they need to demonstrate that they are competent in cleaning and disinfecting. Hand hygiene, gloving, and garbing all need to be proven as a specific competency. Identifying, weighing, and measuring of ingredients. You want to make sure your employees know how to use your scales and your syringes and any sort of equipment you have. Maintaining sterility within an ISO Class 5 area. That would mean things like material handling and things like that. And then also the labeling and inspecting of preparations. So who needs the assessment? All sterile compounding personnel need to undergo this assessment, both prior to initial compounding and at a time period thereafter. So risk levels 1 and 2 have to get a reassessment every 12. The reassessment, again, would include the media fill test and the direct visual observation with the competencies, whereas risk level 3 needs to do that reassessment every 6 months after their initial one. So all risk levels, there might be times when a retraining or reassessment is appropriate. Some examples of that would be if a quality assurance program yields unacceptable results. So if a sterility test comes back as being failed, or maybe you're noticing a trend in your environmental sampling of increased microbial contamination, that might call for some increased training of your employees. If you have any changes in risk levels, or if you observe any unacceptable techniques, you might need to do some retraining. That being said, anytime a person fails any part of the visual observation, such as the hand hygiene, garbing, the aseptic technique, or media fill testing, once they have failed any portion of that, they need to be pulled out of sterile compounding and they need to undergo immediate retraining. So if they failed the hand hygiene section of your visual observation, they would then need to demonstrate that they are able to perform this competency 3 successive times before they are able to start compounding again. So a little bit of detail about the media fills. You will need a policy and procedure for your media fill testing. You want to include the process of your media fill testing, who's going to need to be media fill tested, how you're going to incubate that, who's going to read the results, what temperature you're going to incubate it at, so all sorts of information that needs to go into a policy and procedure. All the media fill testing requirements shall reply with USP Chapter 797, and I will go into more detail on that on one of my future webinars. The process of the media fill test must simulate the most challenging or stressful conditions that are encountered. A lot of people buy the media fill kits, which is okay. Sometimes that is efficient, but they're not necessarily one size fits all. You might need to change the instructions to make the media fill kit to simulate your own challenging process. So if you are doing TPN compounding, for example, you would want your process to simulate that. During the initial media fill testing, so when an employee is first hired, they need to do a minimum of 3 media fill tests, and they need to pass all of them before they are allowed to start sterile compounding. Like I said, we'll have more detailed information on both training and media fills in a later webinar. Section 11 is record keeping, mostly just some additions or clarifications. Training records need to include the dates and the results of the aseptic technique skill assessment and the media fill testing. This would include any sort of retraining that needed to occur. Incubator temperatures need to be recorded if you have one. Certification records need to be kept for both primary engineering controlled and ISO classified areas. You want to make sure that it has that pharmacist review on that certification report, and you want to include any investigations that had to occur due to a deficiency or a failure. If you have a pressure monitoring device, we kind of already talked about that. You need to record those alerts, and all records and reports need to be kept for a period of 2 years, and that's also true for the training records. You only need to keep 2 years worth at a time. So the next section is, or the next slide has section 12, 13, and 18. The reason 18 is on here kind of out of order is that there was just one little change, and I didn't want to waste an entire slide on just one sentence, so I went ahead and added it earlier in the presentation. So section 12 is labeling. There was one label change. If you are compounding hazardous drugs, you need to note that on your label if you have a hazardous drug. For the beyond use stating, there weren't really any changes to that section other than minor clarifications. Again, we added the terminology regarding the microbiological stability, so you need to take that into account when you're assigning your beyond use states. And for the cytotoxic drug system, or cytotoxic drug section, we added one recommendation, which is the use of a closed system transfer device. So this isn't a requirement, it's simply a recommendation. Section 14 is end preparation evaluation. So risk level 1 and 2, there weren't really any changes. Pharmacists still need to visually inspect their final preparation. And you'll notice there was a blurb in there regarding visual inspection. If it were to cause a health hazard to the pharmacist, it is acceptable to use alternate means of inspection. So an example of that would be in nuclear pharmacies. Once the radioactivity is added to the syringe, it would not be safe for the pharmacist to check that syringe. Therefore, they use alternate means of inspection, dose calibrators, in order to confirm that their final CSP is indeed accurate. For risk level 3, we added the addition of USP chapters, so sterility testing must be conducted according to USP Chapter 71, pyrogen Chapter 151, and then endotoxin testing Chapter 85. The rest of this slide isn't any new information, but I just kind of wanted to review it and remind people that these are all still true. So all sterile preparations or a batch from or a sample from each one must be tested for sterility. All parenteral sterile preparations must be tested for pyrogens. If you are assigning a beyond use date that is greater than 30 days for your sterile preparations, you need to do a one-time potency test to prove that that is an acceptable beyond use date. And I wanted to remind everyone about the process for emergency dispensing. So if you prepare risk level 3 sterile preparations and your patient needs that dose for whatever reason prior to the 14-day sterility test results, you are allowed to dispense that product. In this case, you need to call the physician and get authorization for each emergency dispensing. You need to record this authorization and also the need for this emergency dispense within each prescription record. You can't have a physician do a blanket authorization. It needs to be individual for each patient and for each prescription. So Section 16 was created for point-of-care assembled systems. So this is a newer device that has been introduced since our last rule. So we decided to give some guidance. A closed system device that creates a physical barrier between diluent fluids and the drug. So there's a little device that you insert. It has a needle on one end and a spike on the other end. You insert one end into the diluent bag and the other into the drug, and there's a plug in between that keeps them separate from mixing. So it allows the pharmacy to connect them ahead of time, and then it isn't activated until the end user activates it. It breaks open that plug somehow and mixes the drug and the diluent immediately prior to administration. So assembling these point-of-care systems is considered risk level 1 sterile compounding and needs to be done within a primary engineering control within a controlled area. I've listed some examples of some manufacturers in case you weren't sure still what I was talking about. All of these systems, when they're assembled by the pharmacy, shall be assigned 2 different beyond use dates, and both of these dates need to be recorded within the compounding log. So the first beyond use date is going to be for the non-activated state, and this is usually provided by the manufacturer. So the manufacturer usually does their own studies to back up these beyond use dates. If you do not have a manufacturer documentation, the longest you can give the assembled non-activated state, so the drug and the diluent are still separate, is 15 days. You also need to include a beyond use date for the activated state, so in that way the end user knows once they've activated their system how long is a drug stable for, and that would have to apply risk level 1 maximums for that as well. So in that way, if the nurse or whoever activated the point of care system too early, they would know, okay, I have 24 hours before this goes bad. Section 17 is general cleaning and disinfection. So cleaning and disinfection of controlled and buffer areas shall be performed according to Chapter 797. If you are referring to Chapter 97 and you're reviewing the cleaning requirements, and you will notice that 797 does not use the controlled area terminology. They use the terminology of segregated compounding area. So if you're trying to figure out what applies to you and you have a controlled area, it'll be anything that mentions segregated compounding area. So just to review what those cleaning requirements are, I've included a chart here. So ISO Class 5 primary engineering controls need to be cleaned on a daily basis with a germicidal agent followed by sterile alcohol. It also needs to have frequent disinfection throughout the day with sterile alcohol. Examples would be prior to compounding, between batches, and after spills or surface contamination. You would need to disinfect your work area. All counters and horizontal work surfaces need to be cleaned daily. Floors need to be cleaned daily, walls and ceilings need to be cleaned monthly, and storage shelving is monthly as well. And that includes the bins that hold your syringes and drugs and that sort of things, because those can get pretty dirty. If compounding occurs less frequently than the specified time frames, you can go ahead and forego the daily cleaning. So for example, if you only compound on Wednesdays, you don't necessarily have to go in and clean the floors and the counters every single day, but before you start your compounding on your Wednesday, you would have to do all of that required cleaning. You will need a policy and procedure for all aspects of cleaning and disinfection. So this would include things like your approved cleaning and disinfectant agents, your approved cleaning materials, their schedule of use, and methods of application for your cleaning agents. All of the cleaning tools must be low lint and dedicated for use in the controlled or buffer area. You wouldn't want to be using a mop to mop your pharmacy bathroom and then come around and mop your controlled area. So make sure that the cleaning tools are kept for that controlled or buffer area. If you have a germicidal agent that will be used in your primary engineering control and it is not a ready-to-use product and you have to dilute it, you need to use sterile water for irrigation. Regular tap water has a lot of microbes in it, and we don't want to introduce that into our primary engineering controls. So environmental sampling, for the emergency rule, there weren't any changes. For risk levels 2 and 3, we kept the same language, which was applicable environmental monitoring of air and surfaces must be conducted. For the amended rule, we have added a new section, which will be section 18, and that will implement a more in-depth environmental sampling plan. So we will have air sampling as a requirement for all risk levels, which must occur every 6 months. And for surface sampling, risk level 2 compounders will need to do surface sampling every 6 months, and risk level 3 will have to do surface sampling every 30 days. You can do some of the sampling yourself if you have an incubator, or you might need to hire someone to do it for you. And usually certifiers can do that for you. So section 20 is a section regarding remedial investigations. We wanted to give our licensees some guidance on what to do if they were having some issues with microbial contamination. So a remedial investigation is required if any of the required sampling or testing results in a CFU count, so CFU is colony forming unit, that exceeds 797 action levels. So I've included the 797 action levels, so if your environmental sampling shows that you have greater than 1 CFU per 1,000 liters of air per plate in your ISO class 5 area, you need to do a remedial investigation. Same would be true if you have more than 10 for your class 7, or if you have more than 8 for your class 8 environment. For surface sampling, you need to look at the CFU per plate. It's not an additive total, it's just for one plate. And within ISO class 5, you would need, if you have more than 3, you would need to do a remedial investigation. If you have more than 5 in your ISO class 7 area, you need to do an investigation. And then for 8 or worse, it's greater than 100. If the pharmacy detects a highly pathogenic microorganism in any of the preparations, so any failed sterility test that comes back with a highly pathogenic organism, or you find a highly pathogenic organism within your environment or sampling, then you need to do a remedial investigation. So I just want to point out that microorganism identification is not mandatory. However, a lot of our licensees will do this on a regular basis. So if this is something that you choose to conduct, you need to have a remedial investigation if you find that you have a highly pathogenic microorganism. And examples of those include gram-negative rods, coagulase-positive staph, molds, or fungus or yeast. So anytime you have a remedial investigation, any compounded sterile preparations and any ingredients that are part of that investigation shall be quarantined until the results of the investigation are known. And before you start compounding within that area, you need to re-clean and resample the area in order to ensure that you have a state of microbial control. If you do find any of the highly pathogenic organisms, you need to notify the board in writing within 7 days, regardless of the CFU count. So even if it doesn't exceed the 797 action count and you found something in your pharmacy, you need to let us know. Section 21 is just a section about when recalls are required and what to do when that occurs. If your controlled sterile preparation is deemed to be misbranded or adulterated, so maybe you realize you put the wrong label on, the wrong beyond use date, you used the wrong drug, those would all be examples of misbranded or or adulterated, so you would need to do a recall. If your CSP comes back as non-sterile through your end preparation testing, or you have some endotoxin problems, any of those would be a reason for a recall. So when you have to do a recall, you want to notify the prescriber, let him know kind of what happened. If the CSP has the potential to harm the patient, you will also need to notify the patients as well. Anytime you have to do a recall, whether or not it reaches the patient level, you need to inform the board of the recall in writing within 3 business days. You also need to document all activities and investigations that were related to the recall. So that concludes today's webinar. I just wanted to let you guys know about some of the future webinars that I'll be holding. August 17th at noon, I will be having a sterile compounding webinar that gives a more detailed look into garbing cleaning recommendations. And August 25th at 10 a.m., we will have, most likely it'll be over media fill testing and training. And we will have more information for you guys. So if you have any questions, feel free to submit them now, and we will do our best to answer them.
Okay, we'll start off with a couple questions. First, for Kim, we have a question on why was the pharmacy not given more notice of the emergency rule?
And that's a good question. Thank you for the question. I think part of that is because it was an emergency rule. Under Missouri law, there's a procedure for going through an emergency rule, and an emergency rule becomes effective automatically within 10 days. I'd like to encourage licensees to continue to monitor the board's website and also meeting agenda information. The emergency rule was open for public discussion, I believe, in April. And also in May, the board held a conference call and an open meeting to discuss the terms of the emergency rule. Previous versions of the draft were also available on the board's website. But as part of the emergency rule process, given the emergency, the board really wasn't able to give as much educational time beforehand. And once it's approved, it is scheduled for filing, and we then file the rule. In this case, there are several legal hurdles, I guess, requirements that we need to meet. We were informed that the rule was cleared for filing on August 1st with an August 4th effective date. Now, having said that, we understand that it is very hard for licensees, number one, to read the rule, understand the rule, and implement changes in 3 days. So our inspectors have been asked. Katie will be working with everyone to really implement an educational period as we phase in the rule. Licensees should begin working towards compliance, and Katie, the inspectors, the board office, will be working with licensees to help educate you on how to come into compliance. To be quite frank, I think we had a lot of concerns that the inspector will show up. Our inspectors will show up next week and then cite all of these violations in the pharmacy. As we educate licensees, our inspectors will not be writing up compliance with any new requirements as violations right now. Instead, we will be educating licensees on how to come into compliance. Again, that's the purpose of the webinars as well. So while we weren't able to give as much advance notice, we're hopefully able to give more information now that the rule is in place and as we help you come into compliance with the rule. I think the other question that I have, and I think the rest of these are for Katie, was about USP 800 and what the board's intentions were with 800. To be honest, the board wanted to get through the sterile compounding rule first. It was quite the feat, and we've consulted with several licensees. Many of you are on the call. We brought in representatives from hospital. We brought in our nuclear representatives. We had small independent pharmacies, our largest sterile compounding operations, to advise on the emergency rule. Looking at 800, and we understand we have a 2018 effective date. I think the board probably will want to go through a process that allows that level of input as well. The board has not made a decision on adopting 800. We know that it's there. Licensees should be aware that it's effective in 2018. And continue watching the board's agenda to see if and or when the board will adopt 800 in the state. Okay, I think those are the questions for me. We've given Katie a little bit of a break. Excellent job, by the way, getting through a lot of information very quickly. And now I'll let Tom and Katie tag team.
I'll just follow up on Kim's on what inspectors will be doing. Inspectors will be writing on the observation report. We have observations and we have comments. An observation is actually noted as a deficiency. A comment is just a comment that they want to get something across. They will be writing comments on inspection reports on telling you what has changed in the rule and what the new requirements are. So they will be writing something, but it will not be considered a deficiency in the eyes of the board when we're in inspecting in the near future. Okay, we have a question on potency testing. Are you required to potency test each batch, or can you just do one test to validate your process?
So the answer to that question is you can just do a one-time potency test to validate your process. You don't have to send every single batch through for potency testing. Unless your process changes. If there's any variables that change, such as you're using a new formula, new suppliers, new equipment, then you would have to go back and verify your potency again. Okay. For the purpose of cleaning items in the anteroom before bringing into the buffer area, is non-sterile alcohol appropriate, or does it have to be sterile? For our rule, we did specify that it has to be either a sterile alcohol or it can be an equivalent, let me see the wording, equivalent effective non-residue generating disinfectant. So if you have a disinfectant that is equivalent, that doesn't have a residue, you can use that as well, but it has to be either sterile alcohol or that non-residue disinfectant. We have a lot of questions related to how does this affect hospitals and specific hospital scenarios, and I'll try to address that. We have a question on it. We have an outpatient infusion center located in the hospital. If it's within the Department of Health licensed premises, then the compounding, if it's done there at the hospital and then supplying those to the infusion center within the hospital, that would fall under the Department of Health rules. It would not be the Board of Pharmacy. It would not be these rules that Katie's been going over. If the compounding is leaving the licensed premises, then these rules would apply. And so this all goes back again to what's your licensed premises of your hospital according to Department of Health. And every hospital is going to be different on that. You would need to do research to figure out if that infusion center, wherever it may be located on your campus, wherever it might be. It's probably a question that should be asked your inspector to see if they can help you solve that, because every scenario is a little bit different, we seem to find out. We have a question on when is the Department of Health going to have their rules, or does this apply? No, Department of Health has their own rules. They're in the process of revising them. I believe the most current revision references 797. It is not final yet, but I believe the most current draft they have in their rules says sterile compounding should be done in accordance with 797. But again, that is not, they have not been finalized and still subject to change. Okay, we have a question for you, Katie, on is it okay to use tape on the floor to designate a clean restricted area? Yes, that would be an okay thing to do. You just have to be careful if it's something like duct tape. It might, when you're cleaning it over time, it might start to like peel up and it might gather microorganisms. So clean room tape is of course preferred, but if you do use something like duct tape, just make sure that if it starts peeling or coming up that you change it as soon as you can. Do they make specific tape for clean rooms? Yes. Okay. So we would recommend you use that. Right. If yeah, it holds up to cleaning products a lot better. Okay, we have a question again about sterile alcohol and cleaning, wiping down. Supplies must be cleaned with sterile alcohol prior to induction into all ISO classified areas or just ISO 5 areas? The answer is both. So if you have an ISO classified area, you would be wiping it down prior to entering the ISO classified area, and then immediately before compounding, you would also wipe it down. Are dedicated clean room shoes acceptable versus shoe covers? I don't think the board discussed that. So right now our rule says shoe covers would be the only acceptable option at this point.
The rule expects you to document pressure differentials, but you haven't given any guidance on what those ranges should be. What will inspectors be looking for in relation to pressure?
We would probably depend upon what your certifier would identify as being within normal pressure ranges. We don't, since there isn't a certain range that we have identified, we wouldn't necessarily hold you to those standards.
We have some questions about in-use time. They want to know if you're using a closed system transfer device such as facile that has demonstrated longer in-use time for single-dose vials, is that acceptable? The rule talks about if it's been by the manufacturer, and they're referring to the manufacturer of the drug.
Of the medication, right? Because the closed system transfer device could still, if it was contaminated by touch or something like that, it wouldn't prevent any sort of microbial growth from occurring. So it's based upon the manufacturer of the medication, not the closed system transfer device. So it would still be the 6-hour or specific to the drug.
What date should be written on the multi-dose vial? The date you punctured it, time and day you punctured it, or when it expires?
So the date that should be written on your vial should be the date and time that the vial expires. Otherwise, there'll be a lot of confusion, for inspectors and just among your personnel about what does this date mean. So it's better to put the date and time that the vial expires.
More questions on in-use for single-dose vials. With severe drug shortages, is it acceptable to do a risk assessment in lieu of not having critical medications for your patients? The rule does not give any type of exceptions to the process. You either do a 6-hour or you have manufacturer data to back up greater than 6 hours. Someone wanted to know, do all CSPs that do not have an antimicrobial preservative require a beyond use date of 6 hours?
No, the answer to that is no, because remember that the in-use time doesn't affect your final product. It's only the vial itself. So you would still go about your normal beyond use date assignment for your CSP.
This one deals with training. If someone has already been trained prior to the emergency rule went into effect, do they need to retrain with 3 media tests?
No, so the training requirements will be applicable to just new employees that are hired on since the emergency rule, and so that they wouldn't have to do the 3 media fills. They would just have to do, during their next assessment, they would have to do the whole assessment with the visual observations and your specific competencies in addition to just the normal 1 media fill test.
We have a question about if the pharmacy is making their own stock solution to be used, how does the in-use time play into that?
So the in-use time is, so if you have compounded a bulk solution, say you compounded ahead of time, you assign 7-day beyond use date, but you haven't punctured it yet, you've just put it in the fridge for later use, and then you pull that out and you have opened it or punctured it, that is when the 6-hour clock would begin.
So it does apply, even though you made your own. The in-use time will still apply even to stock solutions that a pharmacy has made themselves, but it does not affect the beyond use date of the final product.
Right.
Okay. We have some questions about point of care being assembled outside of the pharmacy. That is not sterile compounding if it's being assembled. So you're just sending the 2 components to a nursing home or other area to be, and somebody else will be doing the assembly. That's not considered sterile compounding, so this rule would not apply. And we have another question about is nurses allowed to do this? And the question, this rule only applies to pharmacies. We don't regulate nurses working out on the floor on how they do what they do. That most likely falls to the Department of Health, either under hospital or long-term care licensing. This couple questions on establishing a recovery time. If they say they have a laminar airflow hood, how should we establish a recovery time?
Usually that is something that is identified by the manufacturer. So if you have your manufacturer manual, take a look in there. If not, maybe if you have an older model, try looking on the internet. You can find a lot there.
And we had another question about someone who always leaves their PEC running, and they want to know, do they have to have a policy and procedure on recovery time?
Yes, in case you have some sort of weird power outage or some sort of strange circumstances like that, it's always a good idea to have the recovery time, and it's required by the rule as well.
We have a question on assessment. If someone works part-time and have been assessed at another location, do they have to be assessed at your pharmacy?
So is the assessment, it should be done at your facility, yes.
Yeah, you can't rely on someone else.
Right.
Because every pharmacy may be different. We have a question, is it possible to save the video with the dialogue? That our recording is both. It will record both the slides that you're seeing and the audio, so you will be able to watch and listen in the future. And we're going to try to speed them up on getting this posted for us. It's actually out of our hands, so but we will try hard to get it up quick. We have a question related to nuclear. They want to know, is a radiopharmaceutical risk level 1 or 2?
It depends what you're doing, first of all, but most of them are usually risk level 1. Some nuclear pharmacies I've been to, they will do some sort of batching activity where they are drawing up, for example, Hespan ahead of time. That would be considered risk level 2. So it just kind of depends on what sort of activity you're doing. If you're not sure, you can send me an email and we can work through that to try to figure it out.
We want to know, is the training part that you talked about the emergency rule or the amended rule?
That is the emergency rule. So it is effective as of now, and then it will remain the same in the amended rule.
Do you have any guidance on where to buy items for the new garbing requirements?
We're not allowed to give vendor recommendations, so I would just work through with your wholesalers and that sort of thing and talk to your friends maybe to get an idea.
On recording incubation temperatures, they want to know if they only use that incubator while they're growing out their media fills. Do they have to record it on days when they're not growing anything?
No, that probably wouldn't be necessary.
We have a question on the nasal, the change in the definition of whether a nasal irrigation is to be sterile. And that is, this person's bringing up, that's not what was previously interpreted, and that is correct. There has been a revision to the board's interpretation, and it actually says now, let me find the actual. It says, this is out of the rule, nasal sprays and irrigations intended for deposit in the nasal passages may be prepared as non-sterile compounds. So if it's being used for just in the nasal passages, it would no longer be considered a sterile compound. Does the end product testing apply to specific patient preparations or only batch compounded preparations? So they're talking risk level 3 here.
It applies to both patient-specific and batch.
But it's for sterility and endotoxin, it's every product, but when it comes to potency, as we said before, it's a one-time as long as you don't change your variables.
Right. I don't think it says.
I have a question on who has to review the pressure differential.
I don't think we have I might have said pharmacist, but I don't think that one. We're looking now to verify. I don't think it has to be verified by a pharmacist. I think it just has to be reviewed. We're
Let's look
looking.
that one up.
Yeah, we're looking.
We'll get back.
We'll get back
look
to
that
you.
one up to make sure we give you the right answer. Are there a list of Board of Pharmacy approved just germicidal agents?
No, like I said, we aren't able to recommend specific cleaning agents. There are generally, I can talk about 2 different types that are generally used, and I will give more details in my cleaning webinar, but there are accelerated hydrogen peroxide products, and another category is quaternary ammonium cleaning products. Both of those are commonly used for clean room disinfectants.
And to go back to our pressure differential, the rule does not say who has to do it, it just says be reviewed daily. I encourage you, if you're using they need to be trained on what they're looking for, so they know that what an appropriate range is, so if it's out of that range, they can report it to the appropriate person. Is a potency test required for each lot with dating over 30 days?
No, as long as you have done the initial potency test in the beginning, that would be sufficient.
When disinfecting supplies into the anteroom, does the alcohol used need to be sterile?
Yes.
And question about tacky mats, are they allowed outside, like in an ISO 8 ante area?
No, that would be an ISO classified area, so you can put them outside of that, so outside of the door leading into your ISO class area. They just create a lot of particulates when you change them, and a lot of times we see that they aren't being changed, so that's why we decided to prohibit them. So you could put it outside the ISO classified, but not anywhere, not even an ISO 8.
Could you please go over the cleaning requirements for a PEC in a segregated area, also the walls, floors, when it's not being used daily?
Sure. So if you so it's a PEC in a controlled area. So if you only compound, say, once a week, you wouldn't necessarily need to go in and clean the floors every single day. But before you go in and compound, you would need to do a cleaning of your floors and a cleaning of your counters and work surfaces, and then, of course, your primary engineering control. Since the walls and the ceilings are monthly, as long as you've done that at least once within that month period, that would be fine. You don't have to do an entire cleaning of the segregated area all on that Wednesday, but it just has to be completed at least once within that monthly time period.
I think we have some clarification. Does that change if the PEC is a RABS?
No, that doesn't change.
Does the number of additives affect the risk level or the beyond use state?
That is something that is more 797 related. That is how they stratify their risk levels. Since our rule isn't adopting 797 at that point, I would say it doesn't affect it at this point.
What kind of documentation do we need to do when reviewing the certification reports?
I would just sign something like reviewed on and who it was reviewed by and the date. And then if you had any clarification, so say your certifier recommended to replace the filter, you could either put an attachment to your certification report about what was done or what questions you asked. It doesn't have to be too complicated. But just a signature and a date would be sufficient enough.
A question about Mini Bags Plus, which is a point-of-care system. They want to know, is it still allowed to let the nurse activate it? And again, this our rule does not apply outside of the pharmacy on who activates, whether and this would be regulated in this thing, it appears to be by a nurse. It'd probably be in a hospital, so that would be Department of Health. But our rule does not specify who can activate. We have a question about the recent FDA guidance on insanitary conditions at compounding facilities. They want to know if the board will be adopting this. That is just draft guidance at the time. No, the board won't. They don't adopt any guidance like that. But everyone may not be aware of that. But the FDA issued some draft guidance last week on that.
I would note that the board will be looking at the draft guidance during its October board meeting, but again, like Tom said, the board doesn't generally adopt draft guidances.
I'll probably cover that in the cleaning webinar. I'm not sure yet.
We have a question on reviewing, and this kind of goes with all these new requirements. They have the specific question is, does the requirement to review certification records make you go back and review the ones you had previously, or does it just apply going forward?
No, it would just apply going forward. I mean, it's never a bad idea to get familiar to reports and, get trends as to what's been happening, but it's only required moving forward.
We have a question on how to specifically label a point of care. Is it okay to say store at room temperature max 15 days, but once activated, use within 6 hours?
Yes, that would be appropriate. That's exactly what you need to include. And then you would need to include both of those dates on your compounding log as well.
For risk level 3 compounding, if you have an employee who's drawing up a dose from a batch vial for the patient, do they have to do aseptic training once a year or every 6 months?
It's not dependent upon the employee. It's
risk level 3.
That's still considered, since you're dealing with a risk level 3 product, they would still have to be doing the every 6-month assessment.
And this goes back to what Katie mentioned previously, that once something is risk level 3, it stays risk level 3 throughout the process. So
right.
At this point, when they're just drawing up the patient dose, it is still considered a risk level, so all the requirements would apply. A question on cleaning supplies. Can the same, I think they're talking about maybe the mops here, can you use the same cleaning supplies for the ante and the buffer area?
Yes, you can. Just remember to keep in mind that when you are cleaning, you want to go from cleanest to dirtiest. So you would do if you are, say, using the same mop for your ante and buffer area, you would start cleaning in your buffer area, since that's the more clean side, and then you would finish off in the ante room. And it wouldn't be a bad idea to change like mop heads in between there either.
Are there board-approved training programs for sterile compounding in the state?
No, there are not. Not that I'm aware of.
No, the board doesn't approve any type of training program. There are training programs nationally out there, but the board would not officially approve any specific program. We have a question about what inspectors have previously been saying on inspection. Prior to this rule, the inspectors were encouraging people to use sterile gloves. Now that the board is issued for risk level 1, the board was prior to this rule, there wasn't a requirement for sterile gloves for any risk level. Now it will be required for 2 and 3. And the inspectors will no longer be making that recommendation. They'll be for risk level 1. There is no requirement, even though it is actually a 797 requirement. The board did not adopt that part of that, so they will not be no longer be doing making that recommendation for risk level 1. I have a question on that. Why do you have to wipe down something? Why can't you just spray it and let it sit?
So this is a common question I get, and the reason is because there are spores within your supplies, so on your IV bags and your vials, that are not killed by just alcohol alone. So if you just sprayed it and let it sit, those spores would still be on your vial or your bag. So it's actually the mechanical process of wiping that allows those spores to be disinfected from that vial or bag.
Can an individual train while waiting for the 3 media fill tests to be done?
So they can be trained, but they shouldn't be doing any sterile compounding for patient use at that point because you haven't received the results of your media fill test. So that if you do the 3 media fill tests, so say you do 1 on Monday, 1 on Tuesday, and then 1 on Wednesday, you wouldn't be able to compound any sterile preparations for humans until 2 weeks have passed since that Wednesday media fill.
We have a question about, do these rules and regs apply to study drugs? And as they're meaning investigational drug studies. There is no exception for investigational studies from the board's rules when it comes to the practice of pharmacy. Now, if it's being done in a hospital for inpatient, then that would fall under Department of Health, but there is no exemptions for investigational drug studies. We have someone asking, will we post the slides? The slides are actually, the handout are posted on our website now. We did add 1 slide after that was posted, so we'll try to get the current version up. But you can find our slides. Just go to our website where it told you how to sign up. There's now a link for handouts.
As Tom is going through final questions, I did want to know, for those who may not know, the board did write and draft an official sterile compounding rule implementation guide. It is, if you haven't received it already, it is being mailed to all sterile compounding pharmacies. If you have a sterile compounding permit on your license, you should receive one. It's also available on the board's website now, and I would suggest that if you haven't seen it, and it summarizes a lot of what Katie talked about today in a very succinct way, please go to the board's website if you do not receive one in the mail, or even if you do, you may want to share it with other licensees.
I have a question on air sampling. Is it preferred or daily air sampling preferred or recommended?
Not necessarily. That's more of an outsourcing type FDA recommendation. It isn't anything that the board discussed, but if that's something you decide to do, that would be up to you.
We have a lot of questions on cleaning, and instead of talking to them now, Katie's going to do our next webinar
Yeah, I will review all these cleaning questions and definitely try to answer them within the next webinar, because I know a lot of this cleaning stuff is new for you guys. So.
And what's the date on that again, Katie?
August 17th. I have it up here. August 17th at noon will be the garbing and cleaning. And then, like I said, August 25th, it says to be announced, but it will probably be media fills and training. And then here are the instructions for filling out the webinar when we're all done. Don't close your web browser window. You will get a pop-up that says the webinar has ended. Click close, and then the survey will then open.
Before we close the webinar, I do want to know, if you have questions, please email the board office, email Katie. Katie is taking phone calls from many of you as well. Her contact information is in the sterile compounding guidance document. It's on the board's website. Again, we want to be a resource for everyone. We understand that this is a change. We want to make sure we provide as much information and education as we can. So please contact us. Let us help you with compliance. Our goal is to assist again so that we are all protecting patients. And I think we're all done, Tom.
Yes. Katie is out this week, so she won't be in her office. So if you email or call Katie, you won't get a response back this week. So just to let you know that. So we're now going to just remind you, August 17th at noon will be the next one. We'll have sign-up posted tomorrow on our website, and there should be another email alert going out tomorrow, also giving you a sign-up opportunity for the next webinar. So we're now going to close the webinar.
Thank you, everyone, for attending.